conclude chapter 11, and then we'll kind of summarize as best we can chapter 12, 12 and 13, kind of get the takeaway about this theory of evolution. And then on Tuesday, we have a big what? Test, unit number 4 exam covering chapters 10, 11, and 12. And at the end of the day today, I will return to you guys your dihybrid cross worksheets so you can kind of look over that over the weekend. Also, don't forget to continue completing your homework through the Connect website. So, okay.
It was kind of like the one with the green and the orange.
Okay.
I didn't understand what it was asking for. There were like 6 questions and it just wanted a number.
Okay.
I couldn't figure out what it was asking.
Okay. Send me an email. I'll look at it.
I need to email before we come back to learn.
Okay. Let me know. It's hard. I can look at it.
And then the dihybrid cross doesn't have all of the word bank doesn't have everything that's needed for it. Is the way that I told my Alex that she didn't want to do it.
I did chapter 11 and we need to put it on.
You guys are good at how to improve this one? Okay. If there's specific questions that I can help, just let me know. But I can't fix it right now. But I can certainly look at it. So if you're having trouble with it, just let me know and I'll do what I can to help you. Alright, but the thing is you're in there doing this, so that's good. That's due on Sunday. Okay, let me.
And that's why I brought more. I think it's more hard because it's like we learned this and then we're going to have one more to do.
On the work part or?
On the actual homework part.
Okay.
That's what I'm having a problem.
Okay, let me reevaluate it. Can you send me an email today? Yeah. If I can change it. I'll see if I can change it since it's open. I may not be able to change it much, but I can delete things now that we're in here covering the material. January when I set it up.
I have a question.
Yes.
For the final, I remember you saying way at the beginning, is it going to replace the test, or is it, if it increases your grade, you'll count?
On the final, in the syllabus, I have it as a required test. And on that test, if it does outperform a low exam, I'll replace that low exam with the final score. Okay. What I think I'm going to do is, if you're coming into this class with an A, I'll give you your average for your. I'm going to make your 5th test optional if you have an A going into the final. Okay. So there's a few of those that'll affect. Okay. But everybody else, you'll take the final exam. I'll make those arrangements. I'll announce that next Thursday about the arrangements for that. But yes, I will replace a low or missing score if it outperforms it.
But only if it outperforms the otherwise.
Right. It's very beneficial. Did I address your question?
Yes.
And you also need email about the homework. Additional items that need to be clarified before we wrap up the semester? Are you happy about that? All right. Well, let's finish up our Unit 4 material so we can go to summer break. Yeah, who is graduating? You guys? Oh, congratulations. Next Friday. All right, so if you remember where we left off on Tuesday, we're going through a little bit of a history of this theory of evolution. We learned that one Gregor Mendel didn't have anything directly to do with evolution, had a typo in your handout, but we learned that Charles Darwin, who's like probably more notable, like to the layperson, if you will, with his contribution to evolution. But the takeaway was that he's not the only guy. He actually didn't come up with this theory of evolution. He didn't come up with that phrase of survival of the fittest and noticed that there was a whole lot of different types of variations of living organisms across the world, which he was able to see and document. And so what he came up with is this concept of natural selection. And what we know is that the evidence out there supports that natural selection does lead to evolution. However, it's not the only causative agent that leads to evolution. So natural selection is one component, but also gene mutations we kind of briefly mentioned in chapter 10, I think. So we should be on chapter on page 5 of your chapter 11 handout. Okay? So some other things that we're going to identify is mutation, non-random mating, migration, and genetic drift. So let's look at these additional components that contribute to evolution. If we look at natural selection, let me see how I have this. Mutation. We know that we've used this word before. We notice there's different types of mutations, like point mutations, etc., where there's like a base, 1 or 2 base substitutions in the DNA during replication, which actually you now know contributes to transcription translational errors. Okay? Or I can call them errors for differences, mutations. And if there's a difference in genetic information, there's going to be a difference in protein expression or phenotype. And if the phenotype changes from generation to generation, we have the ability for some species to now become different, right, species and no longer necessarily interbreeding with one another. So just genetic mutation can lead to these phenotypical changes, which causes what was originally one species to diverge to maybe 2, right, types of separate species, where maybe they become so different at some point that there's a new species. We gave an example, talked about it well on Tuesday. We can see this in high proportion in bacterial reproduction. We had someone have the MRSA, the staph infection, and when those bacteria are mutating regularly, they're dividing so quickly and exposed regularly. So we can really see it in bacteria, but other species who have longer generations, right, it still happens. It just may take hundreds or thousands or more years to actually see that mutation become apparent, where it's obvious to somebody studying a particular species. So the deal with the mutation is not only should it happen, but it's got to be, as I implied, passed on, right, within the population. So not just a point mutation within an organism like a cancerous condition, it's a mutation that happens and then it's carried on in subsequent generations. So not just happening, but also being now expressed in subsequent generations in the population. So natural selection contributes to evolution. Mutation, if it continues in generations, can contribute to this theory of evolution as well. Are you okay on this concept of mutation?
So it contributes to evolution because it can change allele frequency and dynamics.
Yes. So the takeaway is, because the genotype changes, that is the allele, right, which a particular allele would be affected, and then thusly that does affect the allele presentation in a particular population, which is getting into this population dynamics and evolution. You're exactly right. Okay. Thank you for explaining that. Additional reasons why evolution might happen is a concept called genetic drift. So just kind of described as chance, if you will. So it's not like a mutation or something like that. And it's just more of an environmental type of effect, or what we call genetic drift, where some alleles become less frequent in a particular population. And the two primary, and I'm going to apologize, I don't want you to write everything down. I didn't get a chance to update this. So the two primary ways that genetic drift can occur, as I've highlighted for you on page 5 of your handout, include what's called the founder effect and the bottleneck effect. And so during the founder effect, what happens is, in a population, the population becomes usually like so isolated in their environment, or maybe they're hunted, something like an animal hunted, for example, and just the number of reproducing individuals is limited, and so therefore the allele frequency becomes really limited. So almost kind of like inbreeding, if you will, or you might be more familiar with utilizing that terminology. Okay, so there's a surviving population. There's not much diversity in it, so they keep interbreeding, and so it's really limiting the diversity. within a given population. So that population is considered to founder. Conversely, we might have a change in allele frequency due to what's called the bottleneck effect. Okay, so this is more like a kind of a maybe a catastrophic event that happens, or for some reason a bunch of potentially breeding individuals are relocated and they're taken out, and so only those individuals remaining in that area can now interbreed. So it's the same kind of concept, okay, but it's a mass exodus, if you will, of reproducing individuals, leaving just a minute little allele frequency population to interbreed. So in other words, it's just limiting the amount of genetic diversity. We're going to look at this drift. So for some reason, the allele frequencies are leaving those interbreeding populations and not leaving much diversity in there. So mutations, natural selection, genetic drift, okay, can contribute to changing of allele frequencies in a given population. Additionally, many species choose their mate, like you, right? Humans definitely, okay, for the most part, right here in America, we not everywhere, but choose who they're going to reproduce with. We can really see this in different species that have what's called sexual dimorphism, where the males look really pretty and showy and the females not so much, depending on the species. So we see, for example, in this image, a cardinal, right, a red bird. Okay, the male cardinal is the really pretty bird. It's like, oh, cute and whatever. And the female, what does she look like?
I had a male and female right outside on the side of a deep, beautiful. She's just kind of,
They're kind of.
Now this applies to all species, right?
Nope. Not at all. Not at all. I can give some photographic evidence to the contrary. Not at all. But, I mean, there are some, like, so a bacteria. I mean, bacteria aren't going to, no, I mean, they're asexual reproducers anyway, but a lot of organisms are. But, like, say, a plant, it's not going to be like, I can't wait to go get with that tree, right?
And peacock is the same way, right?
Peacock is a great example. Peacock, beautiful tail, peahen. Not so much, right? She's not so flamboyant, right? But
really nice.
do what?
She's really nice.
Friendly. So what that means is a lot of organisms, individuals, can choose their mate based on something that looks appealing or appears appealing, advantageous, really, for the offspring. Okay? And so, a female cardinal wants to pick a red showy male because her offspring will be red and showy and feisty or whatever. Okay? Whatever birds think. Okay? And so there's an advantage to this non-random mating. Okay? So some influences, right? Physically, like physical attractiveness is what we've identified. Behavioral patterns, so you see like bird dances or when you guys hunt, right? How white-tailed deer strut their stuff, during the rut season, whatever, right? Yeah, there's the large racks. Okay, that's obviously no survival. Well, I guess it does have an ability to fight, but it's also a showy. Yeah, and that's why they're fighting, right? And so that'd be a good example of the antlers on deer. Right? Physical location, though, is clearly going to hinder whether or not particular individuals are going to reproduce. There may be the prettiest male cardinal in the world over here in Oklahoma, but in Texas, there may be the best female. But I mean, there's no physical way that those two birds would ever physically mate by their own doing, because they're just physically separated. So I mean, there's and that's kind of almost implied when you think about why some mating choices are not random. All right. And so I already, again, you don't need to write all these down, but what we described was sexual dimorphism where there's a difference between the male and the female. So even though they're the same species, the gender differences, and there's anatomical differences, like in the human, for example, like males tend to have a less mature nature, and the females obviously have memory development, etc. So they feminized appearance versus masculine. So even the humans have this dimorphism. That's apparent that aids in mate choice. So, where we're going is natural selection can lead to evolution. Genetic mutation can lead to evolution. Genetic drift, whether a population founders or has a bottleneck effect forcing on that population. As well as non-random mating contributes to selection of specific alleles. Okay? I mean, the bird doesn't know it's selecting alleles, but it is, for its offspring. Another thing that contributes to evolution is called migration or gene flow. I think your book uses the term gene flow. And so just physical migration of a species between populations contributes to genetic diversity and may lead to eventually different speciation. Humans are a great example, right? You think of where we're going in chapter 12 and 13, we talk about the history of the Earth and a little bit, and we go with a plate tectonic theory where the entire Earth used to be a continual, if you believe that the entire Earth used to be continuous, and it's diverged, all the little plates have diverged, we can see that there are different races of people, okay? But because people can travel, okay, we now have multiple allele combinations, and diversity in those. And the same thing can happen. It doesn't have to happen on a big scale like I'm describing. It can just happen between species of, say, a horned toad hopped in my truck when I traveled from Texas to here to Oklahoma, and it gets out and reproduces, right? So that's a physical, right, relocation, if you will, or genetic flow, right, between populations. Okay, so that's going to kind of mix up the allele frequency. One that you can be more familiar with is just springtime, somebody sneezing back there. What are we catching in our nose? Pollen, you're like, right? Genetic alleles in your nose. So just wind dispersal of pollen is a good way of describing how genes can flow from one population to another. Even though the plant's not moving, it's, you know what it is, getting out there. So it's a good way to illustrate gene flow. So evolution, what is evolution? Great summary. What you said is evolution is a change in allele frequency in a population over a period of time. The scientific theory. We said things that contribute to evolution include Charles Darwin's contribution, which is the concept of natural selection. We also identified it's not the only thing. What else can contribute to evolution of a species? Genetic mutations. Okay, all these things which include genetic mutation. Non-random mating. Migration between populations. The genetic drift. Okay, so if something really severely reduces the interbreeding numbers within a population. So on the founder effect, something has restricted the number of species or individuals, within a population. And so the classic example is that little leopard. Okay. If you think about the leopards, I mean, those animals, they've not gone out to set up different populations. What's happened is they've just been overhunted. Okay? And so there's just a few individuals that can reproduce. And so within the leopard cheetah? I don't know, the zoologist, cheetah. Okay, so the leopard or cheetah? The same thing? Sure. The cheetah that runs the fastest? Yeah. And the cheetah have been overhunted. And so people like their furs and etc. So they've been overhunted. Just catastrophic to the diversity. And so when there may have been hundreds or really thousands of different individuals and however many populations, I don't know how many there were, right? Many populations, we'll just kill them. There's a few that survive, but it's almost like if we were to go to a town, you know, the Smiths and the Browns, Carters, give me a name. Bennett's? Okay, well, okay. I didn't want to give a real name in here. Okay. Well, you'll be the survivor. All right, so something happens catastrophic, and the Browns are killed, and the Whites are killed, and the whoever else I know, but the Bennetts survive. Bennetts only have each other, so Bennetts are making more Bennetts, and those Bennetts make more Bennetts. The diversity, that population just foundered. We call it incest. It's not cool. And the point is, besides the human, just the creepy factor, the point is there's not a lot of diversity. And so the population founders, it gets ill.
I thought the bottleneck effect was the catastrophic loss.
So hunting is not like a single catastrophic event. The hunting is, it's taken over for decades. So it could be multiple events happening over a period of time. The net effect is that population is founded. The diversity is gone. So we can't go back
way to explain the difference.
The effect is the same. Right. The interbreeding population diversity, no. One is something big happens, where the bottleneck, like a catastrophic event.
So the bottleneck is the catastrophic event.
And now you got me wondering myself. So the species will founder upon itself.
That's what I wrote.
Walk myself out of it. The bottleneck, right? Something has happened in the environment to restrict. Now, founder, I think I'm getting it backwards now that I'm walking through it.
So, would disease be the bottleneck of that?
Disease could be a great example of, so hunting is really tragic, but disease could take out a large population of something as well. So, yeah, let me look at, so founder effect, so bottleneck effect is the tragic kind of catastrophic scenario. The founder effect is when individuals kind of move out for whatever reason. And slowly, it could be a disease over a period of time or something like that, or hunting or whatever.
So it could be just catastrophic, but it's not all at once.
Right. In the bottleneck, something pretty sudden. Maybe is a better way to describe that. Whatever it is, disease or earthquake, flood, thunder, snow, fire. Do what? Asteroid. It would not be good. So between the two, the point is the diversity is gone. And as you guys interbreeding, inbreeding, basically. I was like, I don't want to know out your name. Sorry, Miss Bennett. That's going to summarize chapter 11, and I'm going to do my level best to try to present to you in a concise manner chapter 12, if we have time as well, chapter 13, which is going to deal with how additional components of formation of species.
And 13, do you have a test on Tuesday?
We'll see. Let's see how we go.
So, do we still have to do the chapter 13 homework?
Go ahead.
Good question.
Ask me in 45 minutes. All right. So, in summary, here's the takeaway you guys already identified. If you believe in the theory of evolution, that doesn't mean you're a God hater, religious hater, whatever. Okay? I believe in the theory of evolution, and I'm Christian. I believe in Jesus Christ. I believe in God. Okay? Just say it. Okay? So, just because I teach biology doesn't mean that I'm an atheist. It doesn't mean that I hate God. I don't believe in God. I don't believe in... There's no tie to religious belief inside the togetherness. So there's no direct proof to tie them. There may be, but we can't prove it. Okay? So you don't have to hate God, right, to believe in evolution. You don't have to be a scientist. You don't have to teach biology. You just need to understand what we're learning in this class. Scientific theories, there's a whole heck of a lot of evidence, which we'll get to in chapter 12, and just look at it in a logical manner. You don't have to do that to understand. We identified as well as a scientific theory, and we know that scientific theory may be modified if there's additional evidence. Okay? But at this point, there's a whole heck of a lot. And we know, based on our introduction to this class, that science isn't an end-all. We're continually progressing, we're continually learning, we're learning all kinds of different bits of information. So it's just, we're learning. But what we have does support this concept of natural selection and evolution. Okay? Are y'all okay if I say the word evolution now? The heart rates don't go crazy? Sometimes that happens, which is normal. Am I not recording? All right, so let's look at chapter 12, which is going to give us some supporting evidence. We have the idea of what evolution is, and you're like, well, what evidence do you have out there? And so if we look at chapter 12, you didn't have a chance to write down your handout, print your handouts. I didn't put them up till 1 this morning, so you can just take regular notes and then transpose them over to the handouts, which are available on U12 as well. What we're going to do is look at what are the big things that scientists use to support this concept of evolution. We're going to look at what contributes to evolution, physical attributes, genetic attributes as well, and anatomical physical location of species in the environment. As well, we'll talk about different structures, whether they're homologous, analogous, or vestigial. Look at embryonic development and molecular similarities as well. We probably won't talk about this molecular clock. We'll see how it goes. Okay? So the big three things that we're going to talk about today. What about evolution? What evidence is there to support it? Well, one is paleontology. Paleontological evidence, if I can say that. What is that? Study of fossils. So paleontology gives us supporting evidence for this theory of evolution. And up till modern days, I mean, that was the evidence. But now we have additional evidence to kind of support this concept, this theory of evolution. So we have paleontology, which studies fossils. We have physical observations, or anatomical observations, of organisms, whether they be living or deceased, right, fossilized, which is recorded for us. In the adult form, it's structural. But what's really interesting to me is that when a lot of species develop as an embryo, they look the same. Remember, a sperm or a pollen will fuse with an ovum or an egg and begin to make a baby. Little embryo. And if you look at some higher chordates, those animals, y'all studied those, right? Chordates, those that have a what? A spine, okay? They all kind of start out similar. So we'll look at anatomically how some different embryos develop and look similar. And you've got to probably have a good appreciation at the molecular level. There's a whole heck of a lot of similarities between all living organisms, which can help support this concept and this theory of evolution, between our DNA and our proteins in any living organism. Even like ATP synthase. Can you hear that? Okay, that structure is pretty well conserved between bacteria and mitochondria and you inside of your cell. That would be an example. And so that's what we're going to study today. We're going to understand what paleontology is, look at some physical structural similarities and molecular similarities to help support this theory of evolution. Okay to move on? So first we're going to look at paleontology, right? Looking at fossil remains. So the question is, well then, what is a fossil? Okay? How can we define a fossil? What's a fossil?
Remains of an organism
an organism. It may be in rock, but in some form of sediment. And the remains we're going to learn may be partial, or they may be complete. So maybe the entire remains, or it may just be bits of something. And it's got to be pretty old. Okay, so over 10,000 years old, something can be categorized as being a fossil or not. So figure 12.1 in your textbook gives some different examples of fossils as maybe simple as plant structures. We can see a fish, instead of a fish. I'm not sure what this is, but it's like maybe a feather or something like that. Or something really complex like a dinosaur. Partial part of it or all of it. So entire structures are kind of difficult to come across when you look at these big organisms like dinosaurs. But maybe in a rock. The rock's kind of a generic word, so we'll talk about that. But you're right. Okay, so it's a preservation of a species that lived a long time ago, long before you came along. It's studied utilizing this geological time scale. This, too, is one of those things that's debated about the accuracy of guesstimating the history of the Earth. So there's lots of different views on how old the Earth is, and a lot of scientific evidence goes back to how many millions of years ago, so over 4,600 million years ago is a guesstimate of some of the formation of the Earth. And then shortly thereafter, 1,000 some odd years later, million years later, we begin to see some origins of life on Earth. And so we can just work backwards. It makes sense if you think about it, how diversity has arisen throughout the history of life, where we go from more simple, we eventually see species diversify and become more complex. But just because we do have complex development on the Earth doesn't mean that only complex organisms live on Earth. So we can go back to our good friends bacteria and archaea. Not real complex, yet they're living just as fine here in our current time. The deal with fossils is that they do have the ability to give measurement for evolution, for this theory of evolution. So they're able to record and kind of give a time stamp on about when a particular organism survived. And so it gives us some evidence to look at and to measure, to look at the diversity. This Tiktaalik, I'm not sure, came, I don't know, within the last 10 years, kind of identified. And it's believed, and this is where I have difficulty with fossils, is how people interpret it. Like, man, you could just interpret it all day 50 ways. So, I mean, there's a lot of supportive evidence for what some people are saying, but when people say, this is the one that links walking with plot, I'm like, really? You're going to just go jump off the board with that? So, certainly we can see some organisms, we can look at their development and say, maybe they did have some walking type of motion or whatever, but just put your neck out there and say, this is it. I mean, I'm looking at this fossil from hundreds of millions of years ago, and I know without doubt that this is this. I have a little bit of trouble with accepting that, but I can look at this and see that is not a single cell, that was not a single cell bacteria. Okay? I can see that in the chordate, it does have limbs, whether it was using it for swimming or whatever, I don't know. It's a fossil, squished in the ground. But we can make some inferences about how it's built. But we won't definitively, there's no way we can definitively know what exactly it did or when necessarily it was around. So what we're going to look at is how are fossils formed. The methods in which fossils are formed include compression, petrification, impression, cast, and intact preservation. So we're going to go through each of these individually. So we'll first identify compression. So we're looking at how fossils are formed. One way that fossils are formed is by a process called compression. And if you don't have a handout, you can make a reference to figure, it should be 12.4 in your textbook. This is summarized in 12.4. In compression, I mean, what do you think happens? It gets compressed. It gets squished in the ground. Okay? And so And here we have an example of a leaf. So it can preserve part of an organism. Unlikely that it's going to preserve the entire thing. So, for example, this leaf is clearly not from the entire plant which it was associated with. It's part of it, so we can get an idea about that organism. And we can see it probably photosynthesized. We can see some vasculature about it. You can look at the leaf structure, have an idea, see how it's linked to, say, like a rose plant, which we know has a similar kind of ridge type leaf structure. We can make some inferences about it. Those compressions. So what happens is something will settle in an aquatic environment, and then something firm will land on top of it and just physically presses the species. So it's part of it is preserved in compressionism. Am I okay to move on?
So it has to have water to compress?
I don't know if water is required.
So it's just an example?
I think it's an example. If you think about the Earth, water is going to be involved at some point. Where I'm going is it may not have to be a completely like a lake or something like that, but there's going to be a liquid environment when it rains or whatever the case may be. So the environment will be moist, and so that it can compress around that organism. So where I guess I was kind of implying is the volume of water. It doesn't have to be like ocean or lake or something like that. It could just be wherever it's a little bit muddy or damp or something like that. So, good question. Additional questions? So some fossils can be preserved by compression. You may be familiar with petrification. If something petrifies. So what happens is when something is no longer living, what happens is the organic matter, so our carbon-containing molecules, are leached out into the environment, but replaced into the existing structures with minerals. So it's almost kind of like a mold, if you will, kind of like a mold, in that the fossil looks like the original organism, but it's just mineral deposits where the original cells, etc., used to be. And so petrified species organisms are really what? What do you think? Full of minerals, which are real what? We're trying to think of a petrified tree. Do it. They may be able to break. I mean, it depends on how well it was preserved.
Yeah, petrified wood, that's like not the actual wood.
Right, so it's just minerals going in it. So we might pick up a petrified tree right here. Oh, did it? Come on apart? They're heavy. This is where I'm going is. It's heavy. Minerals are heavy. Okay? So it's real heavy. So it's going to be heavier than when the organism was alive. So you've got a bunch of different types of minerals. So how well it's packed or whatever is going to depend on the environment, how much it's been disrupted or whatever the case may be. So whatever you're looking at was solidified. Whatever she was looking at was carbonated for some reason. You going to add something more? So we can compress organisms to preserve them. Some organisms may be petrified in a mineralized environment, replacing the organic matter. Maybe they can affirm something. So as described here, it's kind of like it turns to stone. It's hard. It's solid. It solidifies. So another type of fossil formation is called impression. And this one's going to start out similar to the next type of formation that we're going to study as well. And in this case, an organism will fall into a soft environment, so maybe the ocean, okay, maybe just a muddy environment. And it dies. But as it decays, the environment becomes hardened. And so it's almost like when you drive your truck through the mud, the environment's muddy, it's sloppy when you drive through, the next day it dries out and you can see those tire tracks. It makes an impression. Okay, so fossils can do that as well. And so the impression is not the organism, but it's the remnants of it. Okay, just like The tire track's not your tire, but we know that a vehicle would have driven through that muddy environment. So there's an impression. It's an impression to me. Another type of fossil formation is called a cast, and this looks very similar to this, with a notable exception. A cast, we've got this, we've identified as well that the sediment will harden or leave an impression of the organism. Cast formation is very similar, so that an organism, when it dies, it will fall into a soft environment. It'll form an impression, but in that impression, minerals or something will collect so that the impression is filled. And now we have what looks like the organism, but it's not the organism. It's not direct mineralization replacement. It's just that void being filled. So if you want to go back to the tire track scenario, I could go back after the mud is hardened. I could go back and throw in some fill, like cement or something like that, let it dry, and the cement would be like the cast. Yes. The cast is an impression that's been filled, and that filler has become hardened, and it looks like the organism. This is a great example, but it slipped in my head, so I'm going to share it. Like a cute little ice cube tray. Like say, I wanted to go get OU ice cubes. Right? I could have the ice cube tray. The tray would be like the impression. The ice being formed in there would be the cast. And I could take the ice out. Mine would have look like the OU or whatever. Mine would say OU. I would never call it pistols. Does that make sense? Okay, so the cast is something filling in the void, and the something that's filling in ends up taking on the shape of the organism, but it's not direct petrification formation. Some other cool fossil formation is called intact preservation, and so if you watch Jurassic Park, right, this is the deal right there. Okay. And so we get an entire organism preserved. This is kind of hard to come by. And in order to do this, when the organism dies, it has to be trapped in an environment where there's no oxygen, so the organic materials don't decay or oxidize, right? You get putrid, right? And so, classic example is there's a lot of insects preserved in amber, or previously known as sap. It was from a living tree, becomes hardened and preserved, as well as somewhere where I want to go to look at some intact preservation is the La Brea tar pits. Anybody been there? Yes. Not frozen alive in the La Brea tar pits. Tar. As the organism fall into the tar pit, and the whole organism just sunk to the bottom and is preserved. Does anybody know where that is? Which state? California, near the Los Angeles area. Some species may be frozen intact. Like if you go up north, like really far north to like the Dakotas or Canada, it may be frozen, but not in the La Brea. It's obviously be hot. But in intact preservation, the entire organism is preserved. These are cool because depending on the preservation, just like in Jurassic Park, the DNA may be preserved. We can actually extract the DNA and interpret it, and that's what a lot of people are doing now. They're doing genetic analysis of the fossil. It's pretty neat how it's preserved. So these are the primary ways that fossils can be preserved. The question is then, well, if fossils can be preserved and remain for tens of thousands or millions of years, why isn't there a fossil for everything? Why is there not a fossil record for everything that exists on Earth? Do what? Decay. A little squirrel that I had on there, we were talking about catabolic reactions. Remember him? The little soldiers were standing on it, right? He's going to decay. He's never going to form a fossil. He's just not going to retire, right? Okay. If they die, something's going to may eat it, so something just gets consumed. Meal. Why else might a fossil not be preserved? Maybe the environment doesn't favor petrification or cast formation or whatever the case may be. What else? So burned or destroyed. And so something besides just eating has been destroyed.
Can the animal be too large, or when they can be too large for people?
It may not be too large. If something's too big, it may be earth, like your earthquakes, right? Might be shifted from one location to another. So if it's so large, maybe just pieces get carried off, and so it leaves an incomplete record. So it's hard to interpret maybe what the species is. So think about the opposite scale. Might be too small. Think about a bacteria. Do you think I'm going to be able to find a staphylococcus fossil? I'm not. Somebody might be able to do it. It's pretty darn difficult. What's that?
So that's not something you're going to leave laying around.
Well, if it's fossilized, I mean, it's there. They're there, probably, but we just take a good time to find them. Why else do you think a fossil may not be reported? May not have found it.
Can't dig everywhere.
Can't dig everywhere. Whether you're looking at the poles or deep in the ocean or whatever the case may be. Just not feasible. Okay. Another one that's important to acknowledge is, what do you think is going to be preserved better? Okay, soft tissue, like, say, tissue from your belly, or bony tissue from your back? Which do you think is going to form a fossil better? The bony tissue. So think about all the animals that are not chordates, that don't have skeletal systems in them. So take that into consideration as well. And we identified some organisms just decompose. We said, kind of identify there may be something like a fire or something, or the environment may erode and get exposed and then decay, something like that. We learned that some organisms will be eaten. We identify we can't go everywhere, so something may be deep in the sea you would never find. So there's cool stuff that you don't even know about out there in the sea. So not everything forms a fossil, so that's a hole that a lot of people like to poke at. That will prove this, or can't because of these just natural environmental conditions. The next question that is usually addressed is, well, how can you know the age of a fossil? And there's two primary ways that a fossil age is calculated. The two ways include what's called relative dating and absolute dating. So not like going on a date, right? We're actually trying to determine the age of the fossil. So the terms alone tell you a lot. Relative versus absolute, right? Absolute. Okay, spot on, right? Relative, get an idea, right? Okay. And so the methods of doing relative versus absolute dating are different. So let's look at relative dating. In relative dating of a fossil, we identified it's not going to be specific, it's like a range. Okay, look at a guesstimate of the age of the fossil. And you determine this based on where the fossil was found, looking at the environment, okay, which is called superposition. We kind of mentioned a little bit on Tuesday, but we're just saying, well, obviously, not necessarily obviously, but our inference is that the more deep a fossil is found, the older it is. Okay, so we can look at that just by looking at superposition. Obviously, we know that the Earth shifts, so there's going to be some logic about it, but you can say, well, I found something way, way, way deep in the Earth's core, so it's going to be really old. So you can kind of guesstimate based on other organisms that have been found in that layer. So we can get an idea. Can you see the concept of superposition between this orange versus the light versus the brown basically going through an age? We can also see that erosion can happen, whether it be like in the Grand Canyon, water, like a riverbed or whatever the case may be. So that influences our interpretation. Absolute dating is going to test the fossil directly if there's enough of the specimen, or if the specimen is too precious or too tiny. They can test the environment in which it was found. So it doesn't have to be the fossil itself, in other words, for absolute dating. But what's used are isotopes. And we studied isotopes at the beginning of the semester. These are variants of what? What's an isotope?
A variation of an element.
A variation of an element. Very good. Some examples of isotopes identified in your textbook include C14. What does the C stand for? Carbon. Okay, y'all know that one? Potassium, good job. And these isotopes have what's called a half-life, a known measured half-life. If we look at the half-life of C14, it has a half-life of 5,730 years. And so it says it takes that many years for half of that molecule to decay. Yeah. So the more C14 there is, the younger the younger the species is. And then as it changes, you can I've not done this, but you can interpret the age based on the quantification of the carbon-14 in the sample. Potassium, 40. I can't even mentally quantitate that. 1.3 billion years is the half-life of potassium. And so the way that this, if I use the word half-life, does that mean, is that okay with you, or it takes that long for half of that molecule to degrade? So, if you remember our isotopes, our atomic structure, we say we have protons, neutrons, and electrons. So carbon-12 is our normal, natural atomic element. But what can change to make an isotope? What's different between isotopes? The number of neutrons changes. As we change the number of neutrons, it becomes unstable or radioactive, and it's able to pick up or be measured using certain lab techniques. The question is, what causes this? Okay, when something dies, it's going to decay. And when molecules decay, we know that atoms can be transferred from one thing to another. And so neutrons are pulled from other molecules, and so the carbon can pick up neutrons as other atoms are being decayed out into the environment. And so that's how these isotopes form. Without getting the details, what happens is, just going to give you a snapshot. The sample is collected. Okay. They go through, and it's like, in this case, they're freeze-drying it. They put it through a series of chemical reactions, and then put it into a machine. It's called a scintillator, which measures the amount of radioactive isotopes in that particular organism. So the computer goes through and reads its programs to pick up the radioactive material. Yes?
Is it true that you can test a moth though, and it'll date for millions of years ago? Like you can walk outside and test a moth, and it'll date for millions of years ago.
I don't know. I've never
heard.
I don't know. Just like anything, right? Obviously, there might be nuances to something or some data collection or something like that.
Not St. Lawrence, they had something like that too. It's dated in years ago. Current or
Well, volcanic
or I might be thinking of something wrong, but there's some kind of
something that doesn't make sense.
yeah.
Okay. I'm not sure about the moth. I'll have to look at that and see how the data is collected or what they're doing. So I'm not sure. I can't answer your question, but I'll look into it. and Mount St. Helens was showing some,
Maybe it was volcano. It could have been.
but it's a volcano.
It was like a recent eruption or something.
Well, think
I
of,
could bring it.
not looking at the data, yeah, please do, but not looking at the data, you think about where the volcanic stuff is coming from. The inner core of the Earth, which is how old, right? And so I can see that giving some weird results, even though it's at the surface of the Earth. It's coming from the old Earth, right? The original Earth, from the internal core. So I can see it with a volcano. It doesn't make sense to me with a living moth to give off some ancient age. So I'll have to look into that one. But both of them, if you know what you're looking at, if you could bring that as well, that'd be good. So isotope testing is certainly a valuable tool to try to determine the age of something. All right. Another tool utilized to kind of support this concept of evolution is just the distribution of species on Earth, or which is called biogeography. Okay? So we're just looking at where are particular organisms distributed in the Earth. Like you say, biogeography. Goes back to that theory of plate tectonics and about the distribution of the Earth crust into our individual little continents that we know today. We're going to look at plate tectonics and then the actual physical distribution of species in general. It'll take us a lifetime to look at all species in distribution, but we're just going to get an idea. All right, so if we look at the theory of plate tectonics, the term to describe the original distribution of the continents was called Pangea. Okay? And then over time, the plates have distributed and dispersed, and now we're getting into our current modern day distribution of our continents and our countries that we're familiar with. Okay? And so the theory is that these continental plates move over time. We can have an idea about this. If you go to the edge of any of these areas like California, you know that the Earth is continually shaking, moving. So there's some evidence to support that. And so the theory is that it influenced the past, but also the what? Present. Okay, it's continually influencing our Earth, and thusly have an impact on species distribution throughout our biosphere. So the plate tectonics. Plates move, influences distribution of species. Okay? Now, I already identified some of these scenarios like earthquakes, volcanoes, fracking. Get those plates moving to distribute. But if we look at between, right, so if we look at the original, right, Pangea line, everything was connected. So in theory, we could have some organisms that are on what we would now call one continent. They could move over to another continent. So they could move. And then as they begin to converge, some whole species may have become isolated to what they were originally kind of connected with. This is further supported by fossil evidence. And so if you look at some species that are kind of categorically maybe what you might think of uniquely found in Australia, like marsupials or something like that. They're definitely there. But we find fossil evidence of them in the other continents as well. But they don't exist naturally here now. So there's some fossilized evidence to support this theory of plate tectonics. So if we look at the distribution of species, some species are isolated, and as I identified, some are found on multiple continents, whether that be fossilized evidence or just current distribution under natural scenarios. So in other words, we don't have equal distribution of species, all species in all areas. Some are localized, some are across many continents. summarizing these for you. And 12.9 helps also visualize the distribution of the diversity just between the continents, not even within a given area, but between continents looking at a large scale. There's an area by an explorer called Wallace's Line. It's a deep water trench. So it's not just the ocean, there's like this huge trench in the ocean. So it's given an aquatic and a physical barrier between what looks like just areas that are right next to one another on a map, right? But if you look in the ocean, it's just like huge, deep trench. And so that's believed to allow for this divergence of species. So if you look on one side of it, if you go to the Australia side, that's where you'll find these marsupials. And what over here, we in America, we call it weird animals, right? They're like animals with babies in the pouches and right, all kinds of venomous snakes and all that good stuff, right? And then over here, on the other side of Wallace's Line, we have animals that we're more familiar with, like elephants and bears and all kinds of big critters. And so this line gives a physical separation between the diversity, just between the continents. And so if we go back, just kind of the three tenets that we were looking at some clues to evolution, and we studied, right, we said fossils. Okay, so I'm just re-summarizing. We said fossils, we talked about how they're formed, and we can also see fossilized evidence in our plates, in our continental plates. The second thing that we said gives us clues to evolution are anatomical similarities between different species. So let's now address the anatomical evidence to support this theory of evolution. We're going to look at different types of structures between different species that supports this. Some structures are called homologous, some are called vestigial, and others are called analogous to one another. Homologous is a term that we use to describe chromosomes. That means they what? They are what? What about them is the same? The structure, not necessarily. The structure is the same. The makeup stuff is the same. Okay, so we can have structures as well that look the same, but they may function differently. So if structures are homologous within a species, they kind of look the same, but they may not have the same function. Okay? And the image from your textbook gives a great example of the leg bones. Okay, so obviously we're bipeds, and we walk on 2 feet. And if you compare that to a lion or a seal or a bird or a bat, they're also going to have that same basic bone structure, humerus, radius, ulna, etc. But they'll be built a little bit differently because they function differently in a bat than in a human, whatever. Okay? But we see that similarity. And it's these anatomical similarities that people who support the theory of evolution base part of their theory on. So homologous structures look the same, but they may not function the same. Some structures are vestigial. What does that mean? Here's an example of a snake. It has vestigial legs.
No legs.
Do it?
No legs.
It doesn't have legs, but it has bones, structure of legs, but they're just tiny. And they're not functional, but they're there. They're the exact same orientation. So that says, well, these bones are there. They're not as well developed. They clearly don't have a homologous function. We don't see snakes, thank God we don't see snakes walking around, right? But they do have little hind legs. Do what? Yes. And so, yeah, so you could say, well, snakes might be similar to lizards. Maybe because lizards do have functional limbs. Okay, the snakes, they have the little remnants, if you will. So some structure may be visible, we can see it, but it has no apparent function. Yeah.
Really interesting because we found in Genesis that God curses the snake.
The serpent.
Devil.
Yeah. So they use the word serpent, I think,
I don't know, that's
and I think modern we translate it to a snake. I don't know if serpent is snake, but you're right. And that snakes do have vestigial legs, is what you were saying, right? As well as another thing that kind of contributes to our diversity are structures that are analogous. It's where something it has the same function, but it's built completely different. Like wings. This is a great, your book, I can't come up with anything, but you come up with great examples. Wings of a butterfly are built completely different from the wings of a bird. Birds have bones. Butterflies don't. They use vasculature. Okay, but the whole point is to move. Fly through the air. What are we going to add, Arnold? Or a bat. Okay? A butterfly and a bat. So structures are analogous. They have the same, what? Function, but their structure is different. There is no direct link. And that's interesting. Like, well, why would something need to fly with a completely different structure? Right? So why don't birds and butterflies all have the same structure? Well, they live in different environments. They're built, they grow differently, etc. And so that leads to these analogous structures are believed to be due to this concept called convergent evolution. So the same type of environmental pressures on those organisms kind of force them to adapt, to evolve. But those organisms are completely different environments. Good example from your textbook is like albinism and the lack of pigmentation and photoreceptors. Those animals that live in dark environments, nothing's going to see them. Light's not going to get there. Why waste the energy on pigmentation and visual receptors? We're not going to use them. And so we can see some of those animals that lack pigmentation. So clearly this little shrimp is different than the salamander and that insect. Okay? They're clearly anatomically different, but something about them has evolved similar that they lack any pigmentation. I'm going to use my next 45 seconds, okay, and summarize chapter 12 for you. Okay? Structural similarities, we can see it in anatomical structures. We can also see it in the embryonic development. Make a reference to figure 1215. Fish, mouse, alligator, human. If I were to put these out there, could you tell me which one's which? But if it didn't have a label, a lot of embryos develop similar. Not everything, but a lot of them do, especially when we look at our chordates. There's a lot of similarities. And so there's some additional evidence that there is some genetic, evolutionary linking between many species, even though humans aren't alligators or whatever the case may be. And also, so we've got fossil records to support it. We've got anatomical structures that are similar. And the last thing that I want you to add are those molecular similarities. Our DNA and our protein sequences are very similar between many species. And I gave some examples like ATP synthase, your favorite molecule, right? Or the one illustrated in your book is salivary amylase that allows for you to eat. I will not include chapter 13 on your Unit 4 exam. Thank you. All right. I'll see about removing chapter 13 homework. So just focus on chapters 10, 11, and 12. Is there additional questions? Exam number 4 on Tuesday. So come prepared. Please be awesome. Make an A for me. And I hope that y'all did great on your lab finals, if you've taken already, or Tuesday. Did I say Thursday? Okay. Tuesday. Our next time we meet, we're taking Unit 4 exam. Oh, y'all want your paperbacks too.